Welcome to the nSolver 2 webinar series. In this webinar series, we're doing a series of small webinars on the new features introduced in nSolver 2. In this webinar, I'm going to be talking specifically about the features to do with heat map generation. I'm not going to re-go over any of the other visualization features, only the individual screens specific to heat maps. So I advise you watch the other visualization webinar first. So as with any of the analysis methods, you can use raw, normalized, grouped or ratio data. Here we're going to use the normalized data for our analysis. And you just set up the parameters as described previously. I'm going to exclude the QC samples. In the next two screens, there are some things you can actually do. So you can sort your samples. So if you'd actually put an annotation on the samples, then that can be very useful. So we could sort the samples by that annotation. And you'll see in a later screen how this might be useful. And you can do the same thing with your genes. You can sort them by the annotation. And now we come to the first new screen that's present for heat maps. So when we are running our analysis wizard, we're doing what's called hierarchical clustering, which is a method of cluster analysis which seeks to build a hierarchy of clusters. And what we're doing is the agglomerative approach, which is sometimes called the bottom-up approach. So each observation is going to start in its own cluster, and we're going to form pairs of cluster, and then we're going to merge those pairs of clusters as we move up the cluster hierarchy. They're then going to be presented as results of a dendrogram and a heat map. So I think it's important to understand that what we're doing here is getting you started with the clustering, but the choice of metrics here will change the way the clustering results look. And if you're unfamiliar with some of these terms, you may want to speak with your local statistician. So the first choice you have to decide is whether you want to do z-score transformation, and this is usually done using the z-score of the genes. So what basically happens is that the values are standardized to z-scores with a mean of 0 and a standard deviation of 1. It's a very good idea to transform data with z-scores if the variables being used differ in their variability. Otherwise, those variables with great intensity will influence the clustering, and these could be outliers. So z-score transformation corrects the data internally within a single experiment, and values for individual genes are expressed as a unit of standard deviation from the normalized mean of 0. Correction is done before sample-to-sample comparison. Comparisons across samples across experiments are then performed on equivalently transformed data, and changes in gene expression are then expressed as differences between z-scores. Using this approach, gene expression data derived from different encounter studies becomes comparable across experiments and across labs. So this is usually done with genes. We do provide you the option to use your samples, but this is a very different type of experiment that you're doing. The next choice is the distance metric. And in here, we offer you three choices of distance metric. This is the distance between the clusters as they're being built. The most common method used is Euclidean difference. The advantage for the Euclidean is that it gets most information out of your data, but it's least robust to scaling means and outliers. The next choice is the Pearson correlation, which uses Pearson's correlation coefficient on the log values. It's robust to sample rescaling or shifting, and what this means is it's very good for clustering genes with similar or opposite behaviors. That is, genes that are highly correlated, and those that are highly anti-correlated will be clustered together. And the final choice we give you is the Spearman correlation coefficient, which you can use to cluster together genes whose expression profiles have similar shapes or so similar general trends, for example, increasing expression over time, but whose actual expression levels may be very different in intensity. So you're using rank sorted information here. Once you've chosen a distance metric, you need to choose a linkage method. And we offer a variety of choices: the average, the median, the complete, the centroid, and Ward's minimum distance. The average linkage is the distance between the two clusters is the average of the distances between all points in those clusters. For the median, the dissimilarity between the two clusters is represented by the distance between the median points in those clusters. And in this case, squared Euclidean distance is used rather than Euclidean distance. Complete linkage is the distance between two clusters is the distance between the furthest points in those clusters. For the centroid method, the dissimilarity between the two clusters is represented by the distance between the centroid for the cases in those clusters. And again, we use a squared Euclidean distance here. And then the final method is Ward's minimum distance, which is the distance between two clusters is the distance between the nearest neighbours in those clusters. In many ways, the opposite of complete linkage, where we're using the distance between the furthest points in those clusters. So the final algorithmic choice you need to make is whether you want to cluster on both your samples and your genes, or you want to use an ordered set. So earlier on, I showed you how you could actually order your samples in the samples to be included analysis screen, and how you could order your probes in the probes to be included in the analysis screen. And this means rather than cluster on the samples, for instance, you would just use the order that those samples were in the analysis screen. You could do the same for genes. I'm not going to do either in this particular example. So now we've made all the choices that need to be made to deal with algorithmic components, and there's just a couple of choices left that we'll do with the visualization. Firstly, do you want to use on your sample annotations in the tree view the file name, the treatment group, or both? I have particularly long file names here, so I'm just going to use the treatment group in this example. And the same with the gene. Do you want to use the gene name, its class name, its annotation, its code set name, or its accession number? In this case, I'm going to use the gene name. So now when we press finish, the algorithm does the clustering, and then Java Tree View will open up. Java Tree View has a global window and then a zoomed in window. This is what we're seeing here. And these windows are resizable, so you can have them in any format you want. There are extensive help files and documentation available on the web if you want to read further about Java Tree View. But a couple of key things I found when using this is, firstly, if your global tree is initially not showing up as well as you'd like, you may want to select the fill option from the pixel settings on the menu. This is also a good place to go in and change the color scales. For instance, if you're colorblind, often people prefer yellow and blue. The other thing you'll notice is, remember that I selected to use the sample annotation rather than the file name. So here we can see the sample annotation, and here we see the gene name. These were the options we chose earlier. There are a number of things I can do in Java Tree View. One example is I can select a particular node, and then I can actually flip that. And this is useful because this and this are mathematically the same, but sometimes flipping the nodes can make them look visually more pleasing to the human eye. Once you've finished with Java Tree View, you can quit it, and nSolver will store that analysis for you. You can view the analysis report that shows you the parameters you use to do that analysis. It will show you the samples, and also importantly at the bottom of the file, it will show you any excluded genes. As well as the analysis report, you can recall the entire analysis by clicking on Analysis Data, in which case Java Tree View will reopen so that you can use it. So thank you very much for listening to this webinar. I hope you enjoyed it, it was useful, and I hope you will look at the other webinars on nSolver too, and thank you very much for your time. If you have any questions, please look at the nSolver user manual that was installed when you downloaded nSolver. If you have any questions you can't solve, please contact nSolver support at nanostring.com. And for any general encounter questions, please contact support at nanostring.com. And obviously, for any sales related questions, please contact sales at nanostring.com. Thank you very much for your time. It's been a pleasure to present this webinar.